This lecture covers independent assortment of chromosomes and its contribution to genetic diversity. Here we have two precursor cells, cells that will eventually become gametes, sperm or egg. They undergo meiosis to form 4 gametes, all of which are genetically different from each other. These daughter cells can be used for sexual reproduction to form genetically distinct offspring. Independent assortment of chromosomes is one mechanism that contributes to genetic diversity during meiosis. Let's take a closer look at what this means. We can explore a sample parent cell with 2 genes and how those genes segregate during meiosis. Gene 1 is on 1 chromosome and has alleles capital R and lowercase r, which indicate dominant and recessive alleles. Genetic dominance is discussed in detail in another lesson. The second gene, gene 2, is on another chromosome and has alleles capital A and lowercase a. Remember that meiosis begins with a diploid cell, so each chromosome exists in 2 copies, one derived from this individual's mother and one from the father. Which chromosome comes from which parent doesn't really matter for this example. These have just been randomly assigned to illustrate the point. Each chromosome pair is called a pair of homologous chromosomes, and this parent cell happens to carry a dominant allele of each gene on one of the homologous chromosomes and recessive alleles on the others. Homologous chromosomes align on the metaphase plate during meiosis 1, either in this combination where both dominant alleles, big R and big A, both happen to be on the same side of the metaphase plate, or in this combination where big R is on the opposite side of the metaphase plate as big A. The relative alignment is important because when homologous chromosomes separate during anaphase 1, chromosomes on the same side of the cell will end up in the same daughter cell after the first meiotic cell division. Now, the key to this type of chromosomal segregation is that it occurs randomly, where there is a 50-50 chance that these 2 genes will end up in either one of these configurations. This demonstrates the independent assortment of chromosomes, where the position of one chromosome during metaphase is independent of the position of the other chromosome. Okay, so again, here are the 2 potential outcomes for the alignment of chromosomes during meiosis 1 in our example cell. But this isn't the end of the story. During anaphase 2 of meiosis 2, sister chromatids separate during another round of cell division, forming cells with chromosomes made up of a single chromatid, meaning that if we ended up with both dominant alleles on the same side during metaphase 1, then the chromosome combinations that can be seen in gametes at the end of meiosis 2 will be capital R and capital A in 2 of the gametes we get at the end of meiosis 2, and in the other 2 gametes we would have little r and little a. On the other hand, if dominant alleles ended up on opposite sides of the cell, we'll get 2 gametes with big R little a and 2 gametes with little r big A. So we can see here that a diploid cell with 2 different alleles for a gene on one pair of homologous chromosomes and 2 different alleles for another gene on a second pair of chromosomes can produce haploid gametes carrying 4 different combinations of alleles for those 2 genes. One combination is where both dominant alleles end up in the same cell, big R, big A. Another outcome is where both recessive alleles assort together, little r, little a. Yet another combination is where each cell receives one dominant allele for one gene and a recessive allele for the other gene, in this case, big R, little a. And the 4th combination is also one dominant and one recessive, but now we've switched which one is which, little r, big A. The 4 potential chromosome combinations from meiosis represent a random distribution. These potential chromosome combinations, which are different from the parent cell, increase the genetic diversity of gametes, thanks in part to independent assortment of chromosomes during meiosis. There are other processes that increase genetic diversity even more, but this is a very important one, so make sure you remember it.